The first stage of mitosis is early prophase. Mitosis begins in the nucleus. The DNA begins as chromatin and then slowly condenses. The centromere is replicated.
The next phase is prophase. The chromosomes become more visible and one of the centromeres begins to move to the opposite side of the nucleus. Microtubules begin to form and extend from both centromeres, forming a spindle. The spindle eventually penetrates the nuclear region as the nuclear envelope begins to break up and disappear. By the end of this phase, one sister chromatid of each chromosome has become attached to the microtubules extending from one spindle pole, and the other sister has become attached to microtubules extending from the other spindle pole. During metaphase, the spindle fibers lengthen and shorten, which ends up pulling and pushing the chromosomes. When the microtubules become the same length, all of the chromosomes are aligned midway between the spindle poles. Next begins anaphase. Spindle microtubules begin to slowly separate the sister chromatids and move them toward opposite spindle poles. By the end of this phase, each sister chromatid has now become an individual and unduplicated chromosome. The last phase of mitosis is telophase. It begins when the two clusters of chromosomes reach the spindle poles. Each cluster has the same number and kinds of chromosomes as the parent cell nucleus had. A new nucleus forms around each cluster as the chromosomes loosen up and mitosis is over.